Good afternoon, everybody, and welcome to the Department of Medicine Research and Innovation Conference. My name is Sonia Flores, and I am a professor in the Department of Medicine. I'm also the Vice Chair for Diversity and Justice, and I will be moderating today's sessions. Our second speaker is Dr. Maggie Lam. She is an Associate Professor in Medicine and in Biochemistry and Molecular Genetics. Dr. Lam obtained her BS in Biology and Computer Sciences from the University of Waterloo in Canada, her PhD from the University of Hong Kong. She then moved to UCLA, and after a postdoc and a short faculty appointment, she joined our campus in 2018. Her laboratory develops and applies mass spectrometry and informatics methods to understand the dynamics of human proteomes in health and diseases. Her talk today will focus on mass spectrometry approaches to study cardiac protein expression and kinetics. So take it away, Dr. Lam.
Thank you very much for the kind introduction and the opportunity to share my work here today. So let me just try to do this. Okay. Proteins are workhorses of the cell, and here is a beautifully rendered illustration by David Goodsell of proteins in the human cell. And as you can see, the human cell is populated with numerous proteins that are constantly performing at the various array of biochemical functions. And protein homeostasis is often perturbed in diseases, and as such, most diseases can be characterized by changes in proteins. While the central dogma tells us that genes are transcribed to RNAs and RNAs are translated to proteins to carry out these biochemical functions, the implication is that transcript level dictates final protein abundance level. We, however, now know that there are actually many different layers of regulations between genes and proteins, and also there are many parameters of the cell and of the proteome that govern protein functions. And these parameters can include protein post-translational modification, protein localization, protein turnover, and many others that can be addressed by state-of-the-art proteomics approaches. And today I'm going to share my work on a method to investigate protein turnover and the application of the method on a cardiac hypertrophy model in mouse, as well as in healthy human subjects. So protein turnover is one of the many critical regulatory mechanisms and parameters of the cell. The importance of protein turnover has long been recognized by scientists since 1930s. It is now increasingly recognized as a key feature to control protein quality for proper cardiac functions. And for proteins to be properly controlled, damaged proteins will have to be constantly degraded, and new proteins will have to be made to replenish the protein pool. And these two processes together make up the protein turnover cycle. And disruption in either of these processes can lead to detrimental effects to the heart. So because either protein synthesis or degradation can impact protein abundance. Protein abundance alone cannot inform us of the change in the protein turnover cycle. For example, synthesis and degradation can both be increased at the same time, and their effects can cancel each other out with no observable differences in protein abundance. Another example is that an increase in protein abundance can be due to increased synthesis with normal degradation, or normal synthesis with decreased degradation. And this is the reason why the characterization of protein turnover can inform us of previously unknown molecular parameters that are perturbed in diseases. However, one of the major challenges to determine protein turnover rate is that protein turnover studies are largely confined to single target studies, and there is a lack of technology to derive protein turnover at the omics level. And in our study, we developed a workflow that combines heavy isotope labeling, mass spectrometry, and mathematical modeling to derive protein turnover rates. So one effective way to trace protein turnover is by introducing stable isotope to label amino acids. And these amino acids can get incorporated into proteins. We can model the rate of labeled amino acid incorporation to deduce protein turnover rate. A particularly useful stable isotope that we chose as our protein tracer is deuterium, which is a stable isotope that can be introduced in drinking water. And the reason we chose heavy water deuterium oxide over other stable isotope is because of its ease of administration, its proven safety record going back decades, and it's compatible with clinical studies in humans. So what happens to the amino acids and proteins inside the cell when we introduce deuterium into the system? So immediately following heavy water administration, the cell will start making amino acids with the deuterium isotope. And as labeling progresses, these labeled amino acids will begin to be incorporated into proteins during protein synthesis. And as labeling continues, the majority of proteins will be labeled and the labeling process reaches the steady state. And for us to determine the protein turnover kinetics from isotope labeled proteins, we will have to measure two experimental parameters. The first parameter is how much label have retained in the organism, and we call it the enrichment level. For us, this is achieved by measuring deuterium in plasma of the organism being labeled. We first isolate plasma from blood and perform deuterium exchange with acetone, then subject the sample to GC-MS analysis along with the concentration curve of deuterium. Then we can derive the original amount of deuterium that has retained in the organism. The second parameter we have to measure experimentally is the incorporation rate of the labeled amino acids into proteins. And for this, we employed a shotgun proteomics approach. We extract the proteins from the tissue sample, then we digest the proteins with enzymes into peptides and subject these peptides to mass spectrometry analysis to obtain a set of experimental mass spectra on the peptide. So how exactly do we derive protein turnover from mass spectrometry data? This is a mass spectrum from our shotgun proteomics experiment showing many peptide peaks. It tells us the mass to charge ratio of each peptide as well as their relative abundance to each other. And on the surface, we see one mass to charge ratio for each peptide. But if we zoom into this peak circled in red, we can see that the single peak we can see that this single peak is actually composed of many different peaks. And as we know, these are the isotopic peaks of the same peptides. And this isotopic pattern we see here is from naturally occurring isotope. The cell doesn't care whether it is C12, C13, hydrogen, or deuterium. It sees a carbon, it sees a hydrogen, and it grabs them to make proteins. So before labeling start, we see this naturally occurring isotopic pattern. When we begin introducing deuterium at time 0, we will see naturally occurring isotopes only still because the cell has not had time to make proteins with the labels yet. And as labeling progresses, at about a few hours after the introduction of the label, we're going to start to observe a change in the isotopic pattern. And this is because the light isotope begins to disappear and the heavy isotope that we artificially increase is starting to be made into proteins. And as labeling continues, the pattern will continue to change until labeling reaches the steady state. So the change of the isotopic pattern is modeled by this equation to determine protein turnover kinetics. And we are modeling the rate of change of A, which is the abundance of the first isotopic peak measured by mass spectrometry, normalized by the entire isotopic pattern. A decrease in A reflects the increase in isotope label in peptides. And the rate of change of A with respect to time is equal to the turnover constant times the difference between maximally decreased A, given the amount of deuterium enrichment, A max, and A at time t. And A max is equal to the initial A times the combinatorial probability of peptide having only light isotope, which is a function of the deuterium enrichment level that we measured by GCMS and the number of labeling site of the peptide. So the equation becomes this here on the right. But what I have been showing you is with the assumption that the deuterium enrichment is at a constant level. However, in a more realistic situation, the deuterium enrichment slowly increases as we feed the organism heavy water each day, which also turns out to be following first order kinetics. So our final model is that we incorporated this change into the equation below and obtain this final equation here. And so we applied our developed workflow to a model of induced cardiac hypertrophy in mice, and we induced hypertrophy by using isoprotanol. We administered isoprotanol for 14 days while supplying the mice with heavy water and collected their heart tissue and blood at multiple time points. We also looked at the reverse remodeling of cardiac hypertrophy by withdrawing isoprotanol after 14 days and only then starting to feed the mice heavy water. So from this set of experiment, we derived the individual turnover rate of 3,000 individual cardiac proteins. The measured protein turnover rates were highly diverse and spanned more than 100-fold in dynamic range, with half-lives ranging from less than 1 day to greater than 3 weeks. In the normal heart, the average cytosolic protein species turned over about 10% per day, and the average mitochondrial proteins turned over approximately 5% per day, which translate into half-lives of approximately 6.5 and 15 days, respectively, for the cytosolic proteins and the mitochondrial proteins. We also observed protein complexes exhibiting diverse turnover rates among each other, but turned over synchronously within themselves. The complexes reacted to hypertrophy stimulation differentially. For example, the 60S ribosomal protein complex turned over increased prominently under hypertrophy, as represented by the red bar, being more separated from the normal condition represented by the gray bar than some of the other complexes. And cardiac hypertrophy also led to differential turnover in different metabolic pathways as a whole. Glycolysis proteins and ribosomal proteins turned over substantially more than proteins in fatty acid oxidation and amino acid metabolism. So we next examine the individual subunits of electron transport chain complex displayed in this image. We characterize the subcomplexes with a heat map. The elevation of turnover rates was reflected by the orange color, and the faster it is, the more orange it is. As we can see, the beta subcomplex and the ethanol subcomplex exhibited more elevated turnover rates during cardiac remodeling. And from the literature, we are informed that the heart switches from utilizing fat to glucose during remodeling, and our data perhaps provide an explanation to this mystery. It supports that an alteration in the turnover rates in glycolytic proteins may be underlying this manifestation of the disease phenotype, the switching from utilizing fat to glucose. And so one possible scenario for this long unexplained observation could be that the glycolytic proteins are regulated by their turnover rates instead of their abundance. And from these experiments, we identified about 60 disease signature proteins through turnover dynamics. These include proteins known to be mutated in diseases such as fibrillin and desmin, proteins with known induction in heart failure, such as annexin, but also proteins with unknown involvement in heart diseases. And we have been very fortunate to be able to translate this workflow and platform to human subject studies. We have labeled 10 healthy human subjects for 14 days with heavy water and collected their blood at multiple time points during the labeling. We obtained the turnover rate of 500 plasma proteins from these subjects. And these turnover rates span a range of 70-fold from albumin with a half-life of 23 days to IGF-2 that has a half-life of 8 hours. So in order for us to manage and process the large amount of mass spectrometry data, we also developed a software tool with Java named Proturn to enable the analysis of heavy water labeling data. So basically, Proturn takes in protein identification file and the original mass spectrometry data as input and generate turnover rate constant as output for us. And the workflow of the software analysis involves finding peptide peaks of all identified proteins in the raw mass spectrometry file, integrating the LC peak areas of each peptide isotopic signal and constructing time series with peptide isotope abundance from each time point, and finally fitting data time series to kinetic curves to calculate the protein turnover rate constants. And in order for us to reach the broadest audience, we implemented a graphical user interface to our software. And with this interface, the users can easily submit their inputs for integrating and curve fitting operation with a few clicks. And they can monitor progress of the analysis in real time and look at the results within the same software and compare data between samples. So lately, we've been paying more attention to protein alternative isoforms and not just canonical proteins. We found that alternative isoforms exhibit different turnover rates, which suggests that the isoforms are under very different physiological regulation in terms of synthesis and degradation. And here I'm showing an example of isoform-specific turnover in the case of a protein called pyruvic kinase. And so what we observed was that the turnover rate of the isoform-specific peptides, the peptides that are uniquely belong to only one particular isoform represented in red, showed significantly different turnover rates compared to peptides that are shared among the isoforms. And last but not least, we are also interested to know how changes in isoforms may lead to changes in the protein structures that may ultimately affect protein stability and hence their turnover kinetics. And here I'm showing an example which are displaying the differences in structures of a proteins between its canonical isoform versus its alternative isoform overlaid on the canonical with the help of AlphaFold. So the blue translucent part of the right structure may be what we think are missing from the alternative isoform compared to the original isoform. And we're looking to determine how that may affect the stability and function of the protein. So in summary, we developed a method based on stable isotope labeling, high resolution mass spectrometry, and mathematical modeling to measure protein turnover kinetics. We derived individual turnover rates of about 3,000 mouse proteins in the mitochondria and cytosol and about 500 human proteins in the plasma. And we discovered alternative isoforms from the same gene can exhibit distinct turnover rate. And we are now working to gain insights into how alternative splicing can reshape the proteome through structural prediction using AlphaFold. And with this, I'd like to acknowledge my lab members, especially Wes, who is leading the effort in protein structural prediction using AlphaFold, as well as our lab's collaborators and funding support from NIH. Thank you.
Thank you, Dr. Lam, for a very illuminating talk. So Before people can actually write their questions on the chat or the Q&A boxes, I do have a question. Again, I mean, this is not my area of expertise, but so you are providing the heavy water, and the label is being incorporated into all of the molecules in the cells, including proteins. But my question is, how do you account, and I'm assuming that the majority of these proteins are probably more soluble, or you solubilize them. But what about the rate of incorporation into protein, not complexes, but rather insoluble proteins like the neurofibrillary tangles in Alzheimer's disease? So these proteins actually essentially are more in a denatured form. And what is the efficiency of incorporation of the labels into proteins that essentially are not participating in the physiology of the cell?
Well, so the efficiency. It really depends on whether the proteins are actively being degraded and being made. I mean, from what we know, that if we supply a certain amount of the deuterium oxide into the cell, let's say if we provide the drinking water as deuterium oxide to the mice, and then we measure whether there's changes in how much they utilize the deuterium versus how much they just utilize hydrogen as the molecule to make proteins. What we see is that, let's say if we supplied about like 5% of deuterium to the mice, then we can actually see that it's being incorporated to up to 5%. So it's pretty close to how much we supply to the mice eventually. Well, of course, it depends on how long we supply it to. So whether the labeling reaches a steady state of labeling. So I'm not so certain about specific proteins that may be less soluble, but from what I understand, as long as they're being like actively synthesized, they should take in the label as part of their proteins. Yeah.
Yeah, no, I mean, again, I was wondering because a lot of these proteins, again, a lot is known in the central nervous system, right? So you get these tangles, and you have these proteins that presumably are denatured and form inclusion bodies. And again, I was wondering if is the label incorporated as efficiently, and if it is, do they serve as a depot of even though slowly turned over, there's still sources of amino acids that are labeled to proteins that are not part of these denatured complexes?
Yeah, I'm not very certain about the answer of that, but I think it's a very thoughtful question, and we haven't looked at like proteins from every organ yet, and we mostly focus on cardiac tissue, and so I think that's something that's very interesting for us to look at in the future. Yeah.
And I have another question. Again, I mean, you were looking at mitochondrial proteins and
Right.
a lot of these metabolic proteins. And my question is, if you now, and you were stressing the system by creating and causing the hypertrophy, but if you were to stress the system by, for example, increasing oxidative stress, that we know will modify proteins, will that have an impact on the rate of incorporation of the labels? Because we do know that oxidized proteins are, most of them are turned over faster. Can you comment on that? I mean, how do you address that?
Oh, so yeah, so I think it would certainly affect the rate of incorporation if they are being, let's say, affected in the protein synthesis rate and protein degradation rate. And in terms of oxidative stress, I think for us, for the hypertrophy model, we actually see a lot of differences for the incorporation, because when the heart is being in hypertrophy, it actually synthesizes a lot more proteins. And so we see a huge change in that. But for oxidative stress, I would still think that there are going to be a lot of changes, but maybe not as pronounced as in the model of hypertrophy. Yeah, but definitely we will see some changes if we apply it to an oxidative stress model.
Okay, thank you. Are there any other questions from the participants? I don't see any other questions, so let me check the chat. I don't see any other questions, so I guess this concludes your talk. Again, thank you so much for teaching us or teaching me something new, and I really appreciate it. And again, thanks to both speakers today, and hope you have a great rest of the day.
Thank you very much.